So we're going to talk about Chapter 11. So this is the nervous system, fundamentals of the nervous system. So really, the nervous system follows homeostasis. It is all about feedback. So there is gathering information from internal and external sensors and bringing it back to the central nervous system, where you can integrate and make sense of it and interpret that sensory input. And then you have to send instructions out to the effectors to deal with it. So we talk about sensory input, integration, and motor output. Those are the 3 steps of how the nervous system works. So you have some sort of a sensor and nerves, wires going to the central nervous system where integration happens, decisions are made, and then actions are taken with the motor output. So we look at it, and it's really kind of just a convenient division, but we talk about the central nervous system and the peripheral nervous system. And really the central nervous system is the brain and spinal cord. That's it. Brain and spinal cord make up the central nervous system. Everything else in the nervous system is the peripheral nervous system, the PNS. So the CNS, brain and spinal cord. And this is where integration happens. There is some sensory and some motor originates there and terminates. So sensory terminates there and motor originates there, but it's about integration. We call the sensory side of it the afferent side. Afferent is the same word as affluent. It means flowing towards. There are 2 places that we get information from outside and from inside. Now we divide this up into somatic sensory and visceral sensory. So the visceral sensory is sensors coming from organs. So this would be like baroreceptors in the blood vessels. This would be like monitoring your heart rate, things like that. The fullness of your bladder. The somatic really is skin, muscles, and joints. So the position of your muscles, the position of your joints, and any sensation that comes through the skin is somatic. The motor is the efferent division. Efferent and exit, effluent are all the related words. E, leaving. So the motor transmits pulses from the central nervous system to the effectors. Now, the somatic motor division is voluntary control of skeletal muscles and therefore moves joints and affects our body position, things like that. So the somatic motor division is just what it sounds like. It's very straightforward. The visceral motor division is all about controlling organs and such. And there's 2 parts to this. There is turning these things up and turning these things down. So we call this the autonomic nervous system. And the ANS, the autonomic nervous system, is mostly involuntary. Now, it regulates the smooth muscle contraction, the heart, the glands, things like that. And there's 2 divisions of that. The sympathetic, which is all fight and flight, and the parasympathetic, which is rest and digest. The sympathetic basically turns up most of the organs, except for the digestive system. The parasympathetic turns them down, except for the digestive system. So this kind of breaks it out. So we have the central nervous system going back and forth to the peripheral nervous system. The afferent is in blue, the efferent is in red. The peripheral nervous system is all the nerves, the cranial nerves, the spinal nerves, all these nerves. There is a motor division, right, which is the somatic to the muscles, and the autonomic to the viscera. The autonomic is sympathetic and parasympathetic. The afferent division, the sensory division, is also has a visceral and somatic branch to it. It just doesn't happen to be marked on here. But the blue is this afferent idea. Okay, so we talk about these things and basically the wires. How does this stuff work? There's the histology of the nervous system revolves around two distinct types of cells. One is the neurons. These are the excitable cells. These are the cells that actually have action potentials, the same as muscle action potentials. It's a wave of depolarization that moves down the neurolemma, the cell membrane, letting sodium in and pumping sodium out. So it's a change of electrical, and those are the nerve impulses. And then there are supporting cells called the glial cells. And there's, and when we talked about tissues, we made this list. But in the glial cells, there are 4 different types in the central nervous system: astrocytes, microglia, ependymal, and oligodendrocytes. And in the peripheral nervous system, there are satellite cells and Schwann cells. And we'll kind of look at those each one at a time. So the astrocytes, which are found in the central nervous system, are, astro means star, cyte means cell, so these are star-shaped cells. They support and brace the neurons. They give structure. What they do is keep things in place. Now, they are not static, so they help form synapses between neurons in the central nervous system. They also are responsible for having blood capillaries go to the right spots. So, and they determine the capillary permeability. They're involved with guiding migration. So when I was in school, we thought of them as fairly static things. But research is starting to show that they are actually involved with information processing. That as you form memories, as you make connections between things, you are physically making connections between neurons, and it's the astrocytes that do that. The astrocytes will actually cause 2 neurons that formerly weren't associated with each other to become associated with each other. So it looks like that astrocytes take a part in creating memories. They look like this. They're star-shaped things. And so they're holding on to the capillaries here and the neurons here, and they give structure to the central nervous system. The microglia are really phagocytes. What we don't want is inflammatory responses and macrophages and natural killer cells and such to be in the brain, because neurons can't be replaced. So we use microglia as the phagocytes. They look kind of like starfish, and they engulf any particles that shouldn't be there. Yeah, so they're a protective, defensive cell. The ependymal cells are basically lining all the cavities in the central nervous system. They act pretty much like an epithelium, except that they're not epithelial cells. They are neural cells. So they keep the interstitial fluid, the cerebrospinal fluid, separate. They look like this. A lot of them have cilia, so that moves the fluid along. Looks very much like epithelium. The last one in the central nervous system is something called an oligodendrocyte. And what these cells do is they form the myelin sheaths that insulate one neuron from the other. And they speed up the transmission of action potentials along these neurons. They are very important. These are the cells that are actually attacked in MS, and people with MS have problems with their oligodendrocytes. So the cell wraps and separates the neurons and the axons of the neurons. This is what makes the white happen in the white matter. These things don't exist in the gray matter parts of the central nervous system. Now, in the peripheral nervous system, we have satellite cells and Schwann cells. And they are basically act like oligodendrocytes in that they surround and separate and protect the cells of the peripheral nervous system. Satellite cells surround any cell bodies that are found peripherally. The Schwann cells surround any axons, any of the fibers, and they form a myelin sheath. They guide regeneration of axons, they insulate, and they speed up transmission. So they're kind of a combination in here between oligodendrocytes and astrocytes as far as the peripheral nervous system goes. They kind of perform both functions. Neurons, excuse me, are amitotic. They don't undergo mitosis. They use ATP in crazy amounts, all for the sodium-potassium pump and regenerating the resting membrane potential. So therefore, they need a lot of oxygen and a lot of glucose. Now, they only oxidize glucose because oxidizing fats gives ketones, and oxidizing proteins gives nitrogen, gives amines. And both of those things are damaging to the cells. And because they're amitotic, we don't want to damage them. So it uses a huge amount of glucose. The brain, the central nervous system, is the #1 user of oxygen and glucose in the body, even though it only represents less than 10% of the weight of the body. So therefore, they also have a lot of mitochondria. So the cell body is where the nucleus is, where most of the organelles are. There is this rough endoplasmic reticulum that is involved with making neurotransmitters. It's called the Nysal bodies. It's very dark staining. Yeah, let's just kind of leave it at that. There's an area called the hillock, but we're not going to worry about that. But it basically looks like this. So there's the nucleus, and the nucleolus is where ribosomes are made, so therefore there's a lot of those needed because these things produce the neurotransmitters that are used with every nerve impulse. There are appendages attached to the soma, to the cell body. There are the dendrites, which dendrite means branches. A rhododendron has twisty branches, but dendrite is a branch, and it is receptive. So this long appendage here is called the axon. So really the dendrites are afferent and the axon is efferent. Action potentials and depolarization starts here and transmits this direction. So the impulse direction is this way. We'll talk about the nodes of Rander and how the Schwann cells work in a few moments. So the dendrites and axons. Now, these things get bundled together, and when they're bundled together, like a whole bunch of axons of a whole bunch of neurons together in the peripheral nervous system are called a nerve. The same thing in the central nervous system is called a tract. So if we're looking at the, the optic tract in the brain, that's basically from the optic nerve, which is peripheral, through the brain to get back to the place in the occipital lobe that we process sight. So tracts and nerves. So the dendrites are receptive. They convey electrical signals towards the cell body. They're not full action potentials. They are just depolarization, so they're called graded potentials. It's a little more detailed than we really need, but the axon, there's one axon per cell. Now, the axons can branch, and we call those axon collaterals. But leaving the cell, leaving the hillock back here, is always one. There can be multiple branches of that axon, but at this point, there's only just one. Where the axon synapses with the receptor, so whether if it's a somatic motor neuron, the axon synapses with a skeletal muscle. If it's an autonomic, it will synapse with a either smooth muscle or a gland or something. If it's or cardiac muscle, it can synapse with other neurons. So the axon terminals is where the synapse happens. And they can synapse with the dendrites of another neuron. And it's at the axon terminals where neurotransmitters are released. And these are chemicals that cause a reaction on the receptors of the target cell, the postsynaptic cell. So the axons have action potentials. They're really conducting regions. Then they're the efferent part. Now, stuff has to move along the axons, and these axons can be quite long. Like some of the axons, in the sciatic nerve will, start in the small of your back, and they go right to the end of your big toe. They can be over 1 meter long. So there has to be ways of getting chemicals and proteins and things down and neurotransmitters down towards the axon terminal, and we call that anterograde transport. New mitochondria are made, things like that. They all go where they're needed. And then there's retrograde, which is bringing things back towards the cell body. So this is old organelles that are no longer functional, they're signal molecules. But this is also the way that viruses catch a ride back to the cell bodies, especially the retroviruses. They're called retroviruses because they take advantage of the retrograde movement in the axon. So this would be things like herpes simplex, etc. So the herpes viruses all are retroviruses because they ride this retrograde train to get back to the cell body. And that's a very safe place for a virus to be, because the immune system and the natural killer cells and the T lymphocytes don't kill our neurons because they can't be replaced. So same picture that we just saw. Now, the myelin sheath, which is made up of the Schwann cells, if we go back to here, this whole axon is covered by these Schwann cells. And that collectively is called the myelin sheath. And so that electrically insulates the neurons from each other. So an action potential in one axon doesn't cause an action potential in a parallel one. But it also increases the speed of transmission. So the Schwann cell will wrap around the axon the same way as toilet paper wraps around the cardboard roll, just concentric layers that just wrap and wrap and wrap and wrap. Now, but these are separate cells. Each one of them is separate. In between each of these separate cells is a space, and it's called the node of Ranvier. And this is where collaterals can emerge, but it becomes important because this is where sodium can enter the cell, enter the axon for the action potential. So it wraps and wraps and wraps around like that. Now, not all axons are myelinated. There are some unmyelinated, and these tend to be short, relatively slowly transmitting cells. We use them for integration. We find them mostly in the autonomic nervous system, and they are the short second neuron in the chain. In the central nervous system, there are still nodes of Ranvier, but it's made by the oligodendrocytes, and it's going to form. It will have the same effect. Small neurons don't have this, and that's the gray matter. So again, we have these nodes here. So the white matter and the gray matter, I've already mentioned that a number of times. Okay, so neurons mostly have their cell bodies either in the central nervous system or right next to the central nervous system. So we would never have a neuron cell body out in our fingertip because it's too exposed. It's too likely to be damaged. So we keep it close to the central nervous system, close to the spine and the skull to be protected. So that means we have to have different structures of neurons. So the most common one, the one that we've seen the picture of a few times, is the multipolar. And that's one axon and a whole bunch of dendrites. All motor neurons, whether they're somatic motor or autonomic, all interneurons, processing neurons, are multipolar. By multipolar, it means there's a whole bunch of things going to the cell body. Lots of dendrites, one axon. Bipolar are found in the special senses. So this is the retina. This is olfaction, taste. And these things are have one axon and only one dendrite. Hence, it's bipolar. Most of the rest of the sensory neurons, all of the efferent or the afferent neurons are unipolar. So there is only one thing that goes to or from the cell body, and that process has two parts to it: the peripheral, which goes distally, and the central, which ends up going to the central nervous system. So it tends to be the shorter bit. So the receptors, the dendrites, go onto the peripheral process, and which is really an axon that goes all the way to the cell body and then terminates in the central nervous system by the central process. So it looks like this here, that the receptive endings all transmit along this axon, goes all the way here to terminals, where synaptic terminals, neurotransmitters are released in the central nervous system. And the cell body is fairly close to the central nervous system, and this provides the organelles and things like that. So when we look at this is a multipolar with an axon. This is the bipolar with one dendrite and one axon. And this is the unipolar. These are sensory, these are special senses, this is everything else. So the trigger zone is all around these dendrites. The transmission zone is in pink here, it's all along here. And then the synapses are all in this area here. So it's the secretory. This is where the neurotransmitters are secreted at this end. In the bipolar, a much larger trigger zone and a much shorter transmission, but still secretory being about the same. In the unipolars, trigger zone is here. All of this is transmission and then secretion. The dendrites can be arranged in a whole bunch of ways, but again, these are just some diagrams of some of it. I'm not terribly worried about the details of this. So we've already said the sensory afferent ones and the motor efferent ones carry impulses from the central nervous system to the effectors. Interneurons are between the efferent and the afferent, or the afferent and the efferent, as it were. So this is where we find them. So an interneuron will happen in the spinal cord in this case. A motor neuron happens along there. Sensory neurons go all the way back here, and synapse here. So this and this go together. Unipolar, multipolar, multipolar.